Students, I think there are still many of you are joining, but we'll now not going to wait. Sir, I'll take your minute for your brief introduction, and then we'll see. Yeah. Very good afternoon, one and all, and we have with us Dr. Raman Parkesh. He is a principal scientist at CSIR IMTech Institute, and he did his BSc and MSc from University of Jammu. He completed his PhD from Trinity College, Dublin, and went to Oxford University and the Cripps Research Institute for postdoctoral work. His current research interests include computational medicinal chemistry, chemoinformatics, epigenetics, RNA nanotechnology, supramolecular chemistry for fluorescent chemosynthesis for biologically important ions, solid phase peptoid and peptide synthesis, and development of small molecule probes for various infectious and neurodegenerative disease. He is the inventor in the design and development and commercialization of 3 chemical probe technologies, and he has recently received BIIS Week Award for development and commercialization of first-in-class drug for MDR tuberculosis from Venom. He has published over 60 research articles in reputed national and international journals, and he has 3 books, chapters, and 1 patent in his credit, and he is recipient of many fellowships and awards. So, with very brief introduction, on behalf of Srishti and Honeybee Network, I welcome you, sir. Over to you.
Thanks for this introduction, and it's my pleasure actually to give a talk on this important topic, and hopefully I think over the course of 30 to 40 minutes you will get an idea about what microbes are, what they do to us, and what actually we are. And I hope by the end of this talk you might like microbes. And nowadays because of this COVID situation we kind of hate microbes, but hopefully by the end of this talk you might have a little appreciation for the microbes, and you can see microbes in a different ways. So I will actually start my presentation. What I will show you that what microbes play an important role in, especially in pharmaceutical industry, but I will touch upon a little bit. Microbes are our inherent part. So you cannot separate humans from microbes, and microbes have been associated with us. In fact, they are our part, and that information I think hopefully will come out in this talk. But if you look at the title of this talk, so it's a microbe and it's a pharmaceutical. And if you look at the origins of the words microbes, everyone knows these are small organisms, small life, but most of you might either be aware or may not be aware of a word pharmaceutical. So if you take this pharmaceutical, if you break it down, it's actually 2 words, pharma and ceutical. Pharma actually, in terms of the etymology of the words, pharma means actually it's a lotion, it's a potion, or it's a magical, it's a charm. Nowadays it's called as a drug also, but its initial meaning was some sort of charm, some sort of lotion, some sort of potion which was actually practiced by magicians. And ceutical is actually a therapy or a healing. So in nutshell, if you look, pharmaceutical is a magical potion which can actually heal, and definitely microbes, they do come into that. So hopefully I think you will get to know about them. So I begin this slide with a interesting observation which was made some years back in 1997. So there were actually 2 amateur these climbers. They stumbled upon this mummified body in the Alps mountain, which is in the border between Italy and Switzerland initially, and it was very well preserved. And initially they thought somebody has been murdered there. And they called the police also. The police came, and then they realized they are not actually looking at a murder victim, though it was actually a murder victim, but it's not the recent one. They called the archaeology team. They came up and they dug this body up, and there's a memorial also now in that place where the body was found. And when they realized actually it was a mummified man from 3000 BC. So it's like 5,000 years ago. So, and they were able to actually get so many other, say, interesting things like eggs. And for my interest, what they actually were able to find that this guy was having in his legging, he was having this fungus birch polypore. And when they looked, say later on, it was actually taken to the museum. Scientists were working on that. And by the way, there is a movie also which came in 2017 about this Iceman. And there is a museum about this Iceman. So you can learn so much about the Iceman, about the mass spectroscopy. There are so many things. You look at the online museum, look in the Google, you will find out it's a very interesting things going on. But from my point of view, so this man was having this birch polypore, which is actually a fungus, and this fungus has compounds which are anti-helminthing. And when they actually did a dissection of this man, they were able to see so many other things, but it was actually his intestine was infected with the whipworms, and which polypore is known for the compounds which are having anti-hemorrhagic properties. So it means this, we call it as a Otzi. This Otzi guy was aware of the power of the microorganisms and the compounds which are produced by this microorganism against the disease which he was having. So it means our quest for the medicine was right from so many years ago. So we wanted to actually, obviously we will get ill, we will have some problems, but we had this tendency to think that the natural bounty within that period, or even now also, has the solution for everything, and which is there. So he was aware of that. So in fact, this is the first case of a, we call it as a first aid kit, where you have this bitch polypore lying with him. And by the way, this was actually shot by an arrow. So it was the murder also. So murder were happening that time also. So you can read more in this journal. So what he was suffering from so many diseases. So diseases were there at that time also. You cannot say we were separate from the disease. So he was suffering from so many things. So one of the things was, you can see degenerative diseases, and even arteriosclerosis. So not a very good eater, hopefully. And intestinal parasites also. So that was actually, so humans were aware of the power of these microbes. So if you look at our history in terms of fermentation, so in India also, elsewhere also, the fermentation products were there. For example, 3000 BC, we had somras. Then there were so many other things, and I will actually give you a little bit a demo to the end, like a kombucha, which is a fermented tea. So we were actually aware of the power of these microbes, and we have actually used these microbes for so many things, right from making medicines to the food industry. So hopefully I will show you that, and then I will show you the role of what they do. Okay, so what are these microbes? So probably you might know it. They are too small to be seen. So mostly bacteria, parasites. There are the yeast, some fungi. So if you look at the scale, so mostly they are from 100 micrometer to, say, nanometer level. For example, virus, you need electron microscopy to see them. So they can go up to very small in size. So in terms of the relatively, you need either light microscope or electron microscope to be able to see them, but that haven't prevented their use. And so a lot of studies have been done. We are using them for pretty much everything. I will show you over the course of the talk, so how we have been using them. So if you look, and where are they? Means if you look in our atmosphere, they are everywhere. So you look at the human body, they are in the tongue, they are in the nasal region, they are in the intestine, which is one of the treasure trove of these microbes. Every part of the body, they are in the air up to 100,000 feet. They are in the soil up to 1,700 feet deep, more deeper also, water. So they can go up to the depth of around 7 miles. So everywhere they are. And look in the every industry, So in every organism, they are there. And so if they are there, this is an interesting slide, which actually shows you that if you look at the microorganism and our cells, initially there was an estimate that they outnumber us. For example, there was an estimate that 90% of our body is composed of microbes, but this estimate has been revised, and current standard says that it's almost roughly 1:1, but on a slightly higher side. So it means we have a higher side of microbes. So if you look the ratio, so 43% of our cells, and then the rest is microbes. So we are not even humans. We are actually a mixture of humans and the bacteria and all those microbes. So we call ourselves humans, but actually, if you look, we are not really humans if you go by this analysis. And if you look at in terms of the genes, our human, the genes are like 20,000. But if you take into account all those microbes, so their gene number can range anywhere from say 2 million to 20 million. So if you look at our genome, so we are talking a genome of a size of say, if you take the upper reaches, it will be like 20 million genes, and human has only 20,000 genes. So, and you cannot say that those genes are just lying there and this is just a garbage. No, it means what we are is not because of our genes. It's our genes is there also, but microbe gene also matters a lot. So, but unfortunately, we don't realize the importance of the microbes even still today. We we see microbes in terms of like they are the disease causing. They are the one who are causing, like for example, current pandemic. You can say bacteria is pretty nasty, but if you look, only 0.01%, so a tiny percentage of these microbes are the one which cause the disease, and 99% of those microbes are beneficial to us. So it's a huge number, but that 0.1%, whichever is there, it really has affected us, as I will show you in the next few slides. So there is the negative effect, and this is a very nice graphics. Initially, I wanted to actually show you in a tabular form, but then this graphics is pretty good. So if you look at that in terms of the pandemics, like the so we go to the smallpox, which was very deadly, right to the on these the current pandemic we have. But so the more deadlier are like bubonic plague, and AIDS also occupies that position in that graphics. So this is means most people take this as a scary slide, though yes, it is scary. It actually shows you that microbes, if they go every, they can do a lot of damage. Yes, they affect, but they do have beneficial properties also, which we tend to forget. And if you look in terms of the death, though, say the current COVID-19 is 3.5 million, it's ongoing. Maybe we are lucky we can sort of stop it with the proper measures, and now vaccine has come out. But if you look, the bubonic plague, which was spreading from rats to the humans, it's actually wiped off 35% of the Euro population, and syphilis also. So I will discuss about that. So we are actually more focused on that, and that's how we have been thinking about microbes, that just kill them, but we don't look at the other side of the microbes, what they do to us. They are so important for our body. So I will come to that in the end of my talk. So, and then you can say, well, certain diseases, for example, degenerative diseases, which is caused by the bacteria, shock syndrome, HIV and TB, Ebola, then other viruses, COVID-19. So these are really, so microbes are not really good guys, but unfortunately, we cannot just take these diseases as a mark and then have an all-out war against those microbes, because there are a few bad microbes, but then 99% of those microbes are beneficial to us. So you say solve one problem, but then you create other problems. I will show you later on. But though these diseases actually give us a chance to go for new technologies, for example, COVID-19, by SARS-CoV, it led to the pretty unprecedented development of vaccines and new technology like mRNA-based vaccines and new research. So you can see now the drugs will be faster. So we now have an idea how we can actually accelerate the drug development. So, and going back to, as I told you, that our existence, in fact, we are what we are because of the microbes. So you look in the agriculture, you look in the environmental, food industry, nutraceuticals, and pharmaceuticals. The microbes have an important role to play, and they have really helped us in many ways. I will show you that. For example, just look at the in terms of the human health, insulin, which say that actually diabetes is a lifestyle disease, but earlier on getting insulin was a problematic. You either used to get it from the pigs or from the cadavers, but now you can actually pretty much make it in the bacteria. Interferons, so you might have seen during the COVID times that cytokine storm, interferons are the one which are really helping. Blood clotting factors, streptokinase, and so many other small molecules, medicines, like you look at antibiotics, for example, when you have a COVID or when you have the initial symptom of the COVID, you get erythromycin. These are all produced by the microbes. You cannot make them in a scale, industrial scale, like by synthetic chemistry. So microbes are the one which are helping. And if you look at the lifestyle diseases, for example, you have these statins. So they are again produced by those microbes. So, and maybe some of you might have done research, and every research, whether you're developing small molecules by synthetic chemistry, you need enzymes, you need proteins. So you use polymerase, you use restriction enzymes, you use ligases. They are all being produced by the microbes. E. coli is the sort of toolkit. So microbes are the toolkits of the biotechnology. They are like what we call as hammer and nails. If you want to do any sort of design making proteins, so microbes are the hammer and nails which you need. So they might not look important, but you cannot test a small molecule unless you have the protein. So that's the power of the microbes in the modern biology. And then now we have CRISPR tools, we have genome editing tools, so you can delete genes, you can modify the genes. Synthetic biology is doing a lot, and that too, microbes contribute a lot in that direction. So and food industry. So most of you, while sitting at home, might be enjoying cheese. For example, well, in India, cottage cheese like paneer is more famous, but if you look, the other two cheeses, for example, Kaladi and Kodam, these are Indian cheese from the Jammu region. So that nomadic tribes, they use a lot, and these are fermented products by microbes. Churpi from Sikkim and the other regions of Northeast, that also is one of the cheeses which are made by the microbes. And then you have your yogurt. Every day you take yogurt without thinking what kind of microbes are there. Breads, vinegar, some of the sweeteners. I will show you a demo about a tea, kombucha. Maybe you might have heard about that. So fermented beverages, this is becoming a huge thing now. Because if you look at the life span of the Japanese people, they eat a lot of fermented food. And one of the reasons they live longer as well as they are agile is that they have a very healthy microbiome. And one of the reasons scientists believe is because they eat a lot of fermented food. So, and there is a recent, say, drive towards getting the protein from the microorganisms like bacteria, algae. So you get the protein and the meat. Meat you can now try to make it in the lab, and again, these bacteria and other microbes will be involved in that. So you can have a ethical. You don't have to kill the animals. You can just grow it in the lab using the bacterial power or the microbial power. So that's the impact in the food industry. So you look out for the microbial protein in the near future. So in fact, Louis Pasteur has rightly said that the role of infinite small is infinite large, way back in, say, 19th century. So I would say without microbes, we are nothing. Not because microbes say not only they are an important part of your body, but we use them for making pharmaceutical compounds. We use them in the food. So, and they keep us healthy. They keep us in balance. So I will actually, I kept this as a intentionally blank to actually show you from the beginning where we got the understanding of the microbes and their power. So if you look in, say, for example, 16th century, 17th century, 18th century, we didn't have any idea about these diseases, how they were caused. For example, syphilis, typhoid fever, cholera, and the plague. That's why many people died. We didn't have any idea. What we were thinking, we had this theory called as Myasma Theory, where we were looking that some fall layer is present, and the cause of the disease is from that fall layer. There is some mysterious substances. For example, you can see a cartoon. So from a London Board of Health uh, agency, and you have all the physicians. So these are respected gentlemen physicians, and there is an outbreak, and they are looking for say that say cholera outbreak. They are looking. Some says it's hiding in here, other is climbing on the that stairs and looking it's here. So we had this idea that there is some sort of fall layer, and there is some mysterious substance called miasma, and the disease spreads due to that. Though we had a very good idea about those microbes, but we are not aware that how they are the one which actually cause this disease. And there was this terrible disease, syphilis, which actually wiped 5 to 10% of the European population at that point. So when you have the newly mother which is actually about to give birth, so she was infected, child was infected, and most of the time it will lead to sepsis, and the patient would die, both the child and the mother. So it was a terrible disease at that point. And but so when this disease was going on, there was one gentleman so that was around 1847, a Hungarian scientist, Ignaz. He actually was very meticulous. He looked at, he was actually walking in a hospital. So he was thinking, and just to remind you, at that point, during that point, hospitals were considered to be very clean, and nothing could ever come from hospital. Nowadays, it's not so. In fact, most of the diseases, if you look, hospital is a major contributor. So, but that period of time, up to right up to 19th century, hospital was considered to be very clean. Nothing would come out, no disease. And it was unheard that you can spread a disease from human to human. So you cannot have, this notion was not there. And well-respected physicians and all, they had this notion that there was some mysterious force present somewhere in a place where you can get the disease. But Ignaz was working in a hospital, and he looked at the records there, and he was wondering why so many people are getting sick there in the hospital. Then he did a little bit deeper, and he looked at the mothers who are dying and mothers who are surviving, and then he did an analysis, and he was astonished to see that mothers which are being where like the delivery is being done by doctors and medical students, they are more likely to die by sepsis compared to the mothers where the delivery is by the midwife. Then he just didn't stop there. He looked at why it was the case. So what happened, then you had a hospital there, and the doctors were also studying and working there. So they used to go and do autopsy and come back and straight away deliver the baby. So then he realized that there is something which is transmitting from when they're coming from the autopsy theater to the place where they want to deliver the baby. So something is being transmitted. So he was not sure what it is. But he actually predicted, yes, there is something is coming from there, and that's why it's causing the disease. So what he did is that he actually, in his hospital, he asked that the doctors, if you want to deliver a baby, before you deliver the baby, you have to wash your hand with the lemon. So like in the restaurant, if you go for food in the hand, you get a lemon bath. So doctors actually did that, and the childbirth mortality plummeted from 18% to 2%. and with the more vigorous cleaning, it almost stopped. So, and he wrote this book, The Etiology, Concept, and Prophylaxis of Child Bed Fever. So you can actually find this translation by Cordell Carter. So this is a very wonderful reading because when he wrote this book in 1861, actually everyone ignored him. He was vilified. So big physicians who were working there, he said, it's no way possible that humans can be the transmission of the diseases. How can a disease come from the corpse to the say living person? Hospitals are clean place, but he died tragically. That's a different story. I won't go into that. But just to show you that he was very depressed because he was ridiculed. His book was not taken very seriously. And but then in 1860, Louis Pasteur actually he conducted so many different experiments, and he clearly showed that you cannot have a spontaneous generation of the new life. You need to have um, uh, the organism have to be present in the air, then only you can have a new life. So, but we were not able to see, we still don't see, but now if you put a bread somewhere, you have this mold. You cannot say that it's a spontaneous generation of the mold. No, uh, there is always microorganisms which are present. They are the one who actually cause this disease. So, and in fact, if you look at in the last, scientists had known about the existence of microorganisms since the 1600, but the 19th century medical establishment could not imagine how something so tiny and insignificant could possibly sicken, letten, or extinguish a healthy human being. So it was unimaginable that is something so tiny and so small can actually uh, say get can actually attack us. But it's a fact now. We know that sepsis is a bacterial infection, so we have a pretty good understanding. But then you can see this. So so in 1860s, then synthetic chemistry became more uh, pronounced. We could synthesize molecules, but then came World War First, and during these times there was this guy, Sir Alexander Fleming. He was working in the London, and he lost many of his relatives during the war, and he was consciously looking, or he was deliberately looking how he can kill bacteria. So, in fact, many people died in that war because of infection rather than say gunshot wounds. So he was looking, and his lab was open. He was not a very I would say meticulous person. He was very sloppy in his work, but that sloppiness is the reason we have antibiotics today. So he was actually working on Staphylococcus aureus, and he was growing that bacteria in the agar plates. So then what he will do, he will put anything he will find, put into that bacterial plate and see what will happen. If it is lysis, then he will look back what he had put and then work backward and see what can be there. And if you look, the first antibiotics, most of you will say it is a penicillin, but actually it is lysozyme. So what in 1921, what Fleming did, he had a flu. So he was having this, so his nose was running and his eyes were watery. So what he took a small drop of his nasal drop and put it into the bacteria, and that bacterial colony was lysed, and then he gave it a name as a lysozyme from uh, the mucous membrane of the nose, and he did it also from his eyes. But lysozyme was a very big molecule. Now we know it's a protein, and you cannot actually take it as a sort of oral tablet. So he kept working, and in 1928 he actually was. Many people say this is like anecdotal evidence. Some people say he left the laboratory open and a fungus came, so messed up his agar place, but it lysed the bacteria also. Some people say he actually deliberately put some food or something there where the fungus was growing. Whatever the story is, but the story is that there is a compound produced by the fungus, especially the fungus Penicillium chrysogenium. That compound he named it as a penicillin from the this fungus, and that was able to actually inhibit the growth of the bacteria. So it was killing the bacteria. So that was the first true antibiotics. But lysozyme was the first antibiotics. And say he actually wrote that paper again. It was in journal. I forget that name, but it was some journal of bacteriology in the UK, and he actually said that he had isolated that compound, but it was not taken seriously because you cannot make huge quantities of the compound. Then two gentlemen, Howard Walter Florey and Ernest, they were able to make huge quantities of the penicillin using fermentation techniques. And nowadays we can synthesize also. And this penicillin became a magic drug in the World War II. So 1945, these three gentlemen got the Nobel Prize for the discovery of penicillin. So from there, it just picked up. So we were able to culture the bacteria, some of them. Actinomycetes is a family of the bacteria which nowadays produce 45% of the drug. Fungi, they contribute to 38%, and then you have other unicellular bacteria. For example, now we have these antibiotics, if you look, cephalosporin, penicillin, so many other aminoglycosides. So there's a lot of role of these microorganisms, and we are able to harness the power of these microorganisms. And nowadays there is a crisis because we have not been able to keep up the pace of the discovery of new microorganisms and characterize the compounds. So but if you look in terms of say so many molecules which we have here, for example, from the fungi, Phallosporines, you get these Phallosporines which are nowadays used for immunity, like immune suppressive agents, streptomycin, gentamicin, erythromycins. So these are all from actinomycetes. They are still used when you have the antibiotic infection. So number of different microorganisms are contributing to drugs or pharmaceuticals. I will also tell you one story which you might not have heard, and this has an Indian connection also. So this guy, Surin Sagal, actually, his full name is Surendranath Sagal. So he was born in Amritsar, so which is now a part of the Pakistan. So he, in around 1960s, he went to Canada. So he studied in, his father was having a pharmacy company. So he studied in BHU, but he migrated to Canada. So he was working in a laboratory there. So this is actually, at that time, it was known as IREST Laboratories. Now it's the Pfizer. So Pfizer was earlier name of the IREST Laboratories. So what happened actually, the University of Montreal, they went to this a sacred place in Peru, and there is an island, Easter Island, Rapa Nui. So they have these mounds there, and there is a mystery also how these mounds have been put in there. But they just actually, the residents who were living there, they didn't have many cases of tetanus. So they wanted to see what is there. So they got a lot of samples from each mound. But then the university closed their department and they donated their samples to this laboratory. So Surin Sagal was actually given the job to look out for other compounds. In one of the samples, he discovered that there is a compound, and the compound is rapamycin. And he looked at that it is good for fungal infection, for example, candida. So when you have the candida albicans fungal infection, this compound, you put it and it pretty much takes care of the fungus. But when he went with the data to the company bosses, there was one problem that in addition to it was a very good antibiotics, but then it will say lower your immune levels. So it means it was an immune suppressor. So he went to his bosses and said that I have this very good compound, we should develop it. But they said, no, we cannot do that because it's a immunosuppressive compound. We don't have to do much. So then, so in 1983, this company, Everest, was actually taken over by Wyeth. So again, Surin Sagal went back to, he had a new boss now, because in pharmaceutical company, bosses changes frequently. So I had a new boss. He went to him. He said, "See, I still have the stock of those bacteria, and those compounds are there." So there was, at that time, there was a recently released drug, these cyclosporines, so for the immune suppress, for example, organ transplantation. He said, so we can actually take this molecule and see whether this works as a immune suppressive agent. Then his boss got very irritated. He said, destroy all the stock. So I don't want, just put it in the autoclave and press the button. But Surin Sagal did one interesting thing. He took 2 vials and he took these 2 vials to his home, and he put it in an ice cream pack, and he wrote there, it was written there, not to be eaten. So it was kept in the refrigerator. So then in around 1987, 1988, the company shifted to US, and they again had a new boss. And then Surin Sagal, as usual, he went to the new boss that I had this compound, we should develop it as an immune suppress drug for the transplant patient. This time, luckily, his boss was interested, and they actually he said, but you have destroyed everything. He said, no, I haven't destroyed. As Indians, we keep so many things. So I have this, I have the stock. So he went to home, and he got those vials, produced this compound, and in 1999 FDA approved this drug as a rapamycin. And nowadays many people are making different derivatives of this drug, and there are few chemists in the Roswell Park Institute in Buffalo. They think this drug can be, say, repurposed as a anti-cancer drug or for aging also. So a lot of research is going on, and I think some of you might have seen or might have heard about mTOR or other pathways in cancer. So this mTOR is actually the molecular target of the rapamycin. So this was the drug from the Streptomyces hygroscopicus. So that's an interesting story, how these microbes can contribute to the human health. And so I think most of you, when you look at, say, Hollywood or Bollywood heroes, they look so young, and most of the time you might think what they are doing. Maybe they are eating a pretty good diet, or they are working out. But the secret is this Botox, so where you can get rid of your wrinkles pretty easily, just a small injection, and you are young, 10 years. So Botox is actually also a very powerful, in fact, it's one of the most potent poison from the Clostridium bacteria. And initially it was discovered in the sausages. That's where the name Botulus came from. And then there's a lot of interest was then developed on how you can make this poison or Botox out from this toxin. And it's a secret. There is only Amgen company in US who's doing it. But if you look, it's actually the LD for this lethal dose is 2 nanogram per kilo. So it means if you have a single gram of this toxin, it can kill 1 million people in, say, 30 to 50 hours. And in 2010, actually, FDA approved this botulin toxin for the migraine. So this is how the power it is. It's a poison, but still it has pharmaceutical use for us. And so if you look, the immunotherapy for cancer, we are able to take make antibodies. Normally we do it in the cells, for example, CHO cells, but recently there is a shift towards making these antibiotics in the bacterial, for example, E. coli. So you can make them, you can have either as a soluble protein or as an inclusion body, and then you can do modification engineering and get the interest of your antibody, refolding, optimization. So it's a lot of interest happening in this direction also. And this is another thing, Artemisin. This is actually came from the Chinese traditional medicine. So it's from a plant, but the plant source is dwindling. So you need to produce it. And nowadays, we can pretty much easily produce it in the yeast cell. So what you do is you take the yeast, you tweak, you do metabolic engineering, you can actually delete the genes, you can actually insert the genes, and do yeast what you want. For example, most of you might have used the yeast for making bread, or some people not here in India but outside, they use yeast for the alcohol. So outside, home brewing is pretty much there, but we don't have this thing here. But a lot of people take the yeast and make bread. So yeast, actually there are so many different varieties of yeast. We have only touched 1% of the yeast. 99% of the yeast, other species, we don't really have clue what they do. So, but there might be so many variants in the nature also who can make everything for you. So you just have to find the conditions, grow them. So scientists are doing it now. So you can insert the genes in the yeast and and direct the yeast to make this for me. For example, they have inserted say isoprenoid biosynthetic pathway in the yeast, and that will actually lead to so many of these compounds. For example, artemisins and other drugs, even some of the drugs for the pain. So they are being produced in the yeast. So we have gone further up, and we have actually expressed the ribosomal synthetic pathway in the E. coli. So to make those macrolides, like cyclic molecules like vancomycin. And this is actually now the new thing which is coming up. For example, what you do is that you generate new genes, you generate functional chromosomes. So based on the pathways you have seen in the bacteria, and then you say engineer the yeast, so give a proper nutrient medium, and then you figure out what are the different scaffolds which they are making. Then you can go back and do modification, and go back again to the gene that this modification has to be done. So you re-engineer the pathway, so you can pretty much make up any sort of scaffold. So even cyclic ones can be done. So that's the power of that synthetic biology. Using metabolic engineering, we can modify the pathways. And now we have CRISPR-Cas, where we can delete those chunks which we don't want and insert the chunks. So a lot of things going on. And I also wanted to show you that though my research is on chemical biology, but we have been using microbes a lot. For example, if you look at some of the molecules like NADP or NAADP, which is a second calcium messenger, and most of you might be familiar with NADP, so nicotinic adenine dinucleotide phosphate. But if you replace this amide group with the acid group, so you get the acid version, so NAADP. So it's a very potent calcium messenger, and it's very important target for the virus also, because the Ebola and other viruses, they actually go into this. This is found mostly in the lysosomes and other parts. So they go into this and mature there. So if you can somehow block those the channels, like calcium channels through which NADP acts, you can pretty much stop the entry of the viruses. But I just wanted to show you that if you want to make this compound, so you have this in the blue, and you want to hook on this molecule. So chemical transformation, it's not possible. You cannot make it. So you just put that yeast or Pistoria pistacis or even E. coli, you can have a very good biotransformation. You get those compounds. So that's the power of those microbes. And I have been also working on RNAs. So like, for example, you have muscular dystrophy, or you have Huntington disease or Parkinson disease, where you have misfolded proteins. So you want to study them, so and then develop a drug for that. So you have to make the protein. So where do you make it? So either in E. coli or in the yeast. So for example, MBNL protein or RNA you can make it in the different bacterial system. Riboswitches, you can actually make it in different bacteria, and then you can take those RNA and make nanoparticles. So even we have been working with the MTB, so where we actually take the microbes and test our compounds. So you can see which one is killing. So, and then we are working on different targets on MTV. For example, one of the target is DPRE1. So as you can see from here, cloning and production of recombinant DRP1. So where do you make it? So you use microbes. So otherwise, it's not possible. You can have it, you can give amino acid and make it, but it doesn't work. So microbes are the best thing. And I also wanted to show that where we had a BIIS-9 project. So we get the venom from this snakes, spider, toads. And what's so special about venom? So there are so many different bacterial species, microbes, which are existing in that venomous environment. So they are able to survive because they have special compounds, small molecules, which are there. So we look for the small molecules, and they are useful for different conditions. So for example, there are 5-6 drugs which have been developed from these venoms. So not to say about the least, this area which is becoming more popular. In fact, with the COVID pandemics, everyone is saying we should have more probiotics, we have to improve your immune system. So you have so many different pills available, so many different types of drugs, potions, lotions, which are claimed to be probiotics, which will help you overcome this. And there is a research going on that that these microbes which are in our gut. So if you look at the gut atmosphere, so it's a deep oxygen deprived environment there. Say it's a kind of, you can say, in the middle of the ocean, and it's a very huge sort of place for the bacteria to grow. And the toxins, when you don't have a balance, the toxins and other things which are being produced, they actually go to your brain, and that is the reason scientists now say that is causing the diseases, for example, Parkinson's, Alzheimer's. So if you understand the interface, how the gut microbiota changes, you will be able to design small molecules, or you will be able to design the small antibiotics to overcome these conditions. So there have been some research going on. For example, there's I don't remember the scientist's name, but what he's trying to do is he's trying to raise the mice in a completely sterile environment, and you can see there's the mice which are being generated, they are very fatty, and then he will take the faces of those normal mice and transport into those mice which are generated by in the sterile environment, and those mice are again back to normal. So so if you look Liver Institute in Delhi, they are actually looking for this the faces transplantation. So it's a kind of molecular medicine. So rather than giving a small molecule pill, you actually give a small portion of microbes, take it, and you will be okay. So that will be the next stage, where so you will not take a pill, you will take a tube, and that tube will have whatever name it is. It will be a consortium of different microorganisms, and that will actually cure many diseases. So I would like to thank my students who have been working on different areas and who were instrumental in putting forward this presentation. The director IMTech and funding agencies, CSIR, DVT, BIIS-9, DST, and thank you for listening. And before I actually stop, I want to show you the microbes. So I can actually hand over first. I will show you a demo. I am actually finished, but you bear with me. I have to go to my fridge. I will get something, and then I will show you the colony of microbes and something else. So just bear with me for a second. Okay, so I need someone who can actually, can you see this? Can someone confirm?
Yes, sir. We can.
So what do you see? So do you see something slimy? So if you look, it looks like a sort of messy thing in a pouch. Is it visible?
Yes, sir.
Yes, sir.
So does anyone have an idea what it can be?
Is it a plasma or something?
No. Anyone? So you can see now. You see that soft one on the top. So this is actually okay. You might not be aware, but this is actually a Scooby, and it's actually a symbiotic culture of bacteria and the yeast. And this Scooby is a kombucha Scooby. And what you do is, so it's the microbe. So this is the association. If you take a microscope, you can see it will have yeast. It will have different species of the yeast, bacteria, lactobacillus, so many. Still research going on. So, and if you make a tea, normal tea, like when you make a tea, what you do, you take some tea bags, sugar, boil the water, and then put the tea and sugar and cool, means you can drink hot or cool, but for this to work, you just make tea, normal tea, without putting the milk. So you tea bags and the sugar, and then you put on the top. So when it cools down, you put this on the top. It will generate one more Scooby, so one more baby, and it will do the fermentation of the products. So it actually breaks down the tea and the sugar into organic acids and other things. And this is, if you see this bottle, so this is one of the fermented product, and I have put some ginger also. So can you see bubbles on the top?
Yes, sir.
Yes. So this is carbon dioxide. If I shake this, so you can see a bit more bubbles.
So is it the fermentation going on?
This is fermentation going on. And if I just open it up, you can see the sound also. If I just open it a bit more, it will splash it. So this is the fermentation. And you can drink it. So you can see the sound. So ginger has a lot of this yeast. So you can see the sound. And you can look at how this live this is. So this is from the tea. So this was the demo I wanted to show you. These are the microbes I have at the moment. So this is the power, and you look at the Google. Kombucha is it's a $1 billion industry, and it's popular in India. So I have been using this kombucha say from 2000. I use it for my hobby purpose. I make different teas, and then I do secondary fermentation with the ginger and other fruits like mango. So it has a lot of probiotics. So you can see ginger is pretty good. It works very good with this. So that actually shows you that how friendly the microbes are, apart from few nasty ones. So I am pretty much finished now, and I would like to take questions and hand over to the organization.
Yes. So students, now the floor is open for your queries and questions, if you have any. I expect more questions after this wonderful session. It's really interesting.
Sir, thank you so much, sir. It was very nice, especially the last part where you actually showed us the bottle. I think it really made us understand that these things are very interesting and still can be possible, can be done. They are just not things that we read on paper. So thank you so much, sir. Sir, one question, sir. So when we talk about isolation of antibiotics or antimicrobials from microbes, sir. So how do we go about, say for example, we are looking at one organism, we have a culture of an organism, sir. How do we go about identifying whether it has, whether it can produce any substance that can be potential antimicrobial?
So we beforehand we don't know, but nowadays we can insert the genes in that. So nowadays we know pathways. For example, let me first break it down into two parts. So for example, let's look. You got a microbe like this, and what I do is I do fermentation like this, and then I get rid of that microbes and all. I sterilize it, and then I go to the mass spec, and then I check what is the molecular weight of those. And then from there, we actually work backward to see what kind of scaffolds are there. This is one way. So you can then know what compounds are there, what are the different metabolites which are being produced. Some might be primary metabolites which the bacteria uses. There are also uses, for example, citric acid. Citric acid is used a lot in the food industry. So this is one of the primary metabolites of asparagus. Then you have secondary metabolites. So secondary and primary metabolites, they are being produced in the growth phase, and they are useful for the bacteria or the microbes. Secondary metabolites is actually they are produced at the end or the beginning of stationary phase. So bacteria might produce it, or the microbes might produce it for defense, for pigmentation, or whatever purpose, just like that. So secondary metabolites has a lot of interest because there is where most of the things are useful for us. So what we do is we do this mass spec, then we find the identity of the molecules, and from there backward, we go and find out how we can make it in a larger quantities. Nowadays, what we can do is that if we know the pathway of a particular molecule, how it is being produced, so we can insert that gene into the yeast or the bacteria, and then bacteria or the yeast will produce only that product for us. So that's where the synthetic biology is coming. So we both the traditional way and the where the more modern knowledge is there, where we exactly know that if we insert this gene, it's going to form this. So we do that. But so when we do a traditional way, we don't have say magic wand which will tell us that the bacteria will produce this. You just have to try and figure out what they will do. That's what was being done, where we collect sample from every sort of area, ecological area, for example, from the lakes, from the ocean. We still are not explored much, but the soil, different soil, that's how actinomycetes, they are soil bacteria. And soil bacteria are responsible for 70% of the antibiotics which are being formed. And then just to tell you something interesting, if you look, we have only cultured 1% of the bacteria. 99% of the bacteria are still uncultured. So we have no method to culture them. So we are missing out what molecules they are making, because unless we culture them, they cannot grow, we cannot do fermentation, we cannot actually get the molecules. And there was also a recent, few years back, a paper where somebody actually got the bacteria from the nose of a person, and that was showing antibiotic properties. So it means now you have your colleagues, you just scratch the nose of the colleague, you might get an antibiotic. So I hope this answered your question to some extent.
Yes, sir. Thank you, sir. Sir, also a follow-up question. So when we are doing such things like inserting a new gene, or even when it comes to the extent of genetically modifying the organisms, sir, so is there any strict regulations on it, and has the regulations been imposed more strictly after the CRISPR, the twin incident at China? How has that impacted the regulatory approvals? Has it slowed down research, sir?
Absolutely. See, at your home, you can do pretty much anything. But when you do this research in a lab, we need ethical clearance. We need actually ethical clearance from from our institute. Or if we are submitting a project to the BIRAC, DBT, DST, we need ethical clearance to that. So we actually have to state that what we are doing that, for what purpose we are doing that. It's actually a good question, because See, sometime back, I don't know if you are aware of opium poppy or not. So opium poppy is a plant which is sort of criminalized because the molecules like this opium and say heroin and morphine, these molecules are being produced by the poppy. But there is a way where you can modulate that pathway in a bacteria. And in RNA yeast, and you can make those molecules, like you can make morphine, you can make different those opioids. And that research is actually, in fact, those researchers themselves, they said that it's a very dark area we are going, because humans are very curious. You don't want to end up like a DIY kit from Amazon. You will buy a DIY kit that, okay, this, if you have the yeast species, you have say sugar, just mix some flour like mix some atta, and you will have a morphine bread for breakfast. You will have a what we call as a heroin bread for the dinner. So you don't want to end up like that. So yes, regulations are important, but it is very strictly institutes and the universities and even the where you submit your work, they look very carefully what sort of permissions and those protocols, whether you have followed that. So, and yes, we have to be very careful, because you never know. So there will be some companies which can make those yeast which can actually make morphine. So only thing you have to do is that give the flour and sugar to the yeast, and it will produce a morphine. So you don't want to end up like that. So that is a very important point which we have to consider.
Thank you, sir. Thank you so much for so patiently answering, sir.
Thank you. I appreciate it.
Good afternoon, sir. Sir, like you mentioned about soil microflora, like 1% are cultured and 99% are still unexplored. But the recent practices in agriculture, like we isolate the phosphate solubilizing bacteria or potassium solubilizing bacteria. So I'm just curious that if we isolate some special kind of bacteria and remain separate those microflora from them, so the efficacy will be increase or decrease in like in overall, not only for phosphorus but for other other nutrients as well.
Yes. So yes, it's a good question. So you are knowing the identity of microbe is one thing. For example, you separated from a soil the particular microbe, and you are aware that yes, it forms this. But then you really want to do a detailed analysis. What kind of different compounds it are forming, whether they are useful for the plants. So that's where the interest is happening on the herbicides, or what we call it as a organic those. Insecticides or pesticides. So the problem for us is that we can isolate the organism, we can characterize it, we can actually do a sequencing, for example, 16S ribosomal RNA, we can figure out what it is. But if you want to study it, we need to culture it, we need to actually see what kind of metabolites it forms, what are the pathways. That's where the problem is. So people are developing different properties, they are actually say going into say setting up the culturing in the soil itself. So you have that is the other area which is of interest. So Taxiobactin was one of the organism which was produced. It was there was a new species, it was not culturable. So what they did, actually they cultured in that soil area. So they set up a culture unit in the soil area, and they were able to culture it, and they got a Taxiobactin antibiotics from that. So that is where we have to be innovative and make new methods of culturing.
So one question is, like, I'm working on biomass hash, like not for this or like BIIS, but in general my project. So there is availability of potassium in large extent. So if I want to train a microbes, can we train a microbes like potassium solubilizing bacteria in that way
Yes,
to
yes.
increase the solubility?
you have to first figure out, do this kind of screening, what are the different microbes which are able to uptake that. So you can figure out with the mass spec that much. For example, I showed you this. So there are certain fruits which work very well. They do fermentation. You can actually visibly see ginger is one of them. But there are some which actually they don't do it. The bacteria is not able to utilize that. So you have to do a screening. So you can actually generate a chip, and then you put different bacteria and then different quantities of your compound and see which bacteria is able to overtake it. And by mass spec, you can pretty much do it easily. Multi is pretty good for that.
Sir, if in any time I need your advice, so can I contact you?
Yes, sure, definitely. My email address, you have it, and you can contact.
Sure, sure. So one question is like maybe you heard about xenobots, which is recently developed.
Yes.
So what's your thought on that?
So I would say, see, unless, say, working in this area, in my view, is that unless and until we have a capability to understand really what impact they will make, we have to be careful in going about exploring these microorganisms. So my take, if you want to modify any microorganism, if you want to actually introduce additional genes, additional mutations, you should be also careful about what the consequences can be. So be prepared.
So, yeah, like they are using it for like their plan is for cleaning of oceans or in waste. So
But where it will end up,
that is yeah, like genetic modification chances are there.
modification you can pretty much do. See, you can make so many things, but where it will end up, where it will go, you should be aware. I am not saying that you should not do it. For example, nowadays there is a research that kombucha can be used there also. But what it will do there, that is the additional thing you do. You don't want to end up, say for example, here we have a controlled fermentation. You can stop the fermentation so you don't make up alcohol. So you get organic acid, you stop it. But say suppose you introduce this kombucha into the rivers, you don't have a control over the river and the extent of fermentation. You might have end up making alcohol. So it means then you take water and everyone will be drunk. Everyone takes a bath
Yeah.
there. They will end up. It will be good for them, but then you have to have this possibility.
Thank you so much, sir. Yeah.
Any more questions? Anyway, if you have any question, you can write it to me, or sir, if you don't mind, I'll share your email ID with the student.
Sure. No problem.
Yeah, we can further discuss about their own projects with you.
Sure.
So thanks a lot, sir, for your wonderful session and for your time. And we look forward for your support in such future workshops.
Sure. Thanks. It was my pleasure. Thanks very much.
Thank you.
Bye.
Bye.